anyway, it's nice to speak in my home. So, unfortunately, I can't seem to advance slides. No, those are working? Good. Okay, sorry about that. So, schizophrenia is a neurodevelopmental disorder in which genetic and environmental risk factors interact to lead to the emergence of symptoms, usually in late adolescence or early adulthood. And until fairly recently, drug development has primarily focused on the most florid positive symptoms of schizophrenia. But even when these symptoms can be adequately controlled with medications, a substantial number of patients are left significantly disabled by cognitive deficits. And only as a consequence, only about 20% can work. So cognitive deficits wind up being the strongest predictor of functional outcome in schizophrenia. And since schizophrenia strikes young and affects 1% of the population worldwide, this has staggering economic and psychosocial costs. And so this is obviously a huge unmet medical need. And the problem is we don't understand the neural basis of cognitive deficits, so we lack effective treatments. Now, the NIH has poured countless millions of dollars into efforts to characterize and treat cognitive deficits in schizophrenia. And to my mind, a fundamental limitation to this work is that these studies only measure cognition in cross-section at a single point in time. While this provides a valid snapshot of function, it misses the critical aspects of learning and memory that happen offline, over time, and with sleep. After encoding, memories are not just stabilized like a snapshot, they're enhanced and transformed in the brain. And these consolidation processes happen offline, outside of awareness, and continue over days and even years. And much of this evolution of memory is mediated by sleep. Now, this is not a new idea. This is a Roman rhetorician. It is a curious fact of which the reason is not obvious that the interval of a single night will greatly increase the strength of the memory, perhaps because the power of recollection undergoes a process of ripening and maturing during the night. But subjecting this to empirical study is relatively new. And this is a seminal study by Avi Carney and colleagues that makes a couple of important points. One is that sleep doesn't just passively preserve memories, it actually improves them. This was looking at a perceptual skill. And it's not any type of sleep, but it depends on a certain type of sleep. And in this case, it was rapid eye movement sleep. And in the last 20 years, there's been a virtual explosion of research in the role of sleep and memory. So here's the Avi Carney study, and then this is the cover of Science in 2001, and you can see the explosion of research on sleep and memory. And a lot of this research came from the lab of my colleague and friend, Bob Stickgold, and he's been a real pioneer in this field. So I want to just give you a very brief flavor of what sleep can do with regard to memory. So if I have you read, I'm going to read a list of words: door, glass, pane, shade, house, open, frame, view, ledge, breeze, curtain. What's the gist word that's missing there? Oh, come on. Be brave. Be courageous.
Window.
Window, thank you. Okay, so sleep helps to reduce the rate of forgetting of these study words. But interestingly, for the gist words, sleep actually enhances them. Now keep in mind, these gist words weren't even on the list. So sleep is resulting in perhaps a less accurate memory, but arguably a more useful one. So sleep enhances the gist. And let me just give one more example. So Jess Payne in Bob's lab showed subjects scenes, and there were backgrounds that were neutral, and then there was either an emotional object or a neutral object, let's just say a parked car, in the foreground. And sleep had no effect on memorization of the neutral scenes, either the backgrounds or the foregrounds. And it also did not have a significant effect on recollection of the backgrounds of the emotional scenes, but it preferentially encoded the emotional scene. So it's taking what is relevant. To paraphrase Dan Schachter, memory is less about the past than it is about the future. What information is going to help you avoid danger and optimize decisions going forward. So it's helping to select what is most salient and relevant to survival. So this body of work has led to a paradigm shift. Not all of our learning happens while we're awake. Sleep, rather than being a passive restorative state, is an active period of cognitive functioning that facilitates learning and memory. And this has become so boringly well established that Bob's work has moved from this cover of Science to the cover of Scientific American. So, but what does any of this have to do with schizophrenia? So for that, I have to take you back to the early noughts when I was working here with Randy Golub and others and doing working memory studies in schizophrenia to look at the role of prefrontal cortex dysfunction. And we wanted to use prefrontal cortex dysfunction and working memory performance as outcome measures in drug trials of agents that purported to improve cognition. But before we could do that, we had to be able to show that these measures were reliable. So we did a small test-retest study looking at working memory activation of the prefrontal cortex and performance in a group of patients who were clinically stable and had no changes in medications over the 2 time points. And for controls, when we compare reaction time from test to retest, if anything, they tend to get faster. So they're faster the second time. Patients showed no such tendency. They're not different from 0. It's like they haven't benefited from the first try. And when we look at activation, controls tended to activate about the same amount on both tries. And this was not true for the patients. There was no relationship of activation from the first to the second try. So I was presenting this data at a neuroscience seminar at the Brigham and Women's, and saying that what I thought this reflected was that the patients were failing to hone the optimal spatiotemporal pattern of activation, that they were failing to automate the task. They were approaching it the second time as if it were the first. And after my talk, Bob Stickold came up to me and he said, that depends on sleep. So that resulted in the research program that I'm going to describe today. And I just want to give you what the key points to take home from this are. Patients with schizophrenia have impaired sleep-dependent memory consolidation that may be caused by a deficit in sleep spindles. Sleep spindle deficits implicate the thalamic reticular nucleus, which I'll abbreviate as TRN, and the related thalamocortical circuitry. Spindles and TRN circuit dysfunction are promising novel targets for treatment of cognitive deficits in schizophrenia, and genetic studies are beginning to reveal the mechanisms of these deficits and provide specific molecular targets for treatment. And if you happen to go to sleep in this lecture, part of this is covered in a review. So let me just describe a good night's sleep. Sleep is not a unitary state. A night of sleep can be divided into rapid eye movement sleep and non-rapid eye movement sleep. And so non-REM sleep can be further subdivided into 3 stages. During the night, you cycle through these different stages, going from lighter to deeper slow wave sleep, or N3, approximately every 90 minutes. In the wee hours of the morning, sleep becomes shallower, and you spend more time in REM sleep, which is associated with dreaming, and also in stage 2, or N2 sleep, which is characterized by spindles. Each of these sleep stages is characterized by different patterns of brain activity, and different sleep stages facilitate different types of learning and memory. So we interrogated sleep-dependent memory in schizophrenia using a simple finger tapping procedural learning task called the motor sequence task. And what people do is they put their left hand on the keyboard and they type a sequence, in this case 41324, as quickly and accurately as they can. They do this repeatedly for 30 seconds, as fast as they can. They rest for 30 seconds, and they do this 12 times. And this is what learning looks like in healthy young adults. So they start out fairly slow. Let's see. No, come on. Where is it? What am I doing wrong? Aha. So they start out pretty slow, and then there's a typical learning curve, and they tend to asymptote. It doesn't matter if they learn in the morning or in the evening. When they come back, so there's no circadian effect on learning. When they come back after a night of sleep, there is significant improvement without further practice. And this improvement depends on sleep. So here we test them we train them in the evening. They come back after a night of sleep. There's significant improvement. An additional 12-hour interval, not including sleep, does not result in more improvement. If you test them in the morning and they go through 12 hours of wakefulness, improvement is not significant, but when they come back for a night of sleep, they show significant improvement. So this improvement depends on sleep. So what happens in schizophrenia? This is the control group showing a typical learning curve on the MST. And when they come back after a night of sleep, there's significant improvement. I'm putting up a new y-axis for patients because overall they type more slowly than the controls. But when you superimpose them, their learning curves look pretty much the same, and there's no significant difference in either the amount or proportion learned. But when patients come back after a night of sleep, there is no significant improvement. And this is a significant difference from controls. So they're failing, despite intact learning within the training session, they are showing a specific failure of sleep-dependent improvement. You might argue, well, they tapped overall fewer sequences during training, so maybe they didn't tap enough to show the sleep benefit. So we gave a subset extra practice, so they did an additional 12 trials after the first 12 trials, and when they came back after a night of sleep, they still didn't show improvement. So it wasn't a matter of just not getting as much experience with the task. This failure of overnight improvement in schizophrenia occurred in the context of significantly reduced sleep spindles. So sleep spindles are a defining feature of stage 2 non-REM sleep, or N2, seen in the EEG as brief bursts, 1 to 2 second bursts of 12 to 15 hertz activity in a waxing, waning envelope, hence the name sleep spindles. They sort of resemble a spindle. They are a key mechanism of synaptic plasticity, mediating memory consolidation during sleep. And importantly, in schizophrenia, the spindle deficit is correlated with the memory deficit. The more spindles you had, the more you improved after sleep, suggesting that the spindle deficit in schizophrenia caused the memory deficit. And this causal hypothesis is in line with optogenetic, pharmacologic, and transcranial stimulation studies showing that by increasing spindles, you can improve memory. And Brian Baxter in my lab is now designing a study to see if this can work in schizophrenia. We have also extended the memory deficit in schizophrenia to declarative memory. This is on a word pairs test, and which in previous literature has been shown to depend on spindles. And patients show, despite getting up to a comparable level of learning during training with extra drilling, they show a deficit. They show increased forgetting of the words. This was not true on a visuoperceptual procedural task that the patients and controls both showed significant improvement after a night of sleep, and this is a task that doesn't depend on spindles. So it's a selective deficit and possibly reflecting tasks that depend on spindles. So at this point, you may be asking yourself, are spindle deficits due to antipsychotic drugs, and are they specific to schizophrenia? So this is some work from Fabio Ferrarelli's lab showing that the spindle deficit, you can see these topo maps, and these are, this is power in the spindle frequency range. So we see a deficit in patients with schizophrenia compared to controls, but not in a medicated group of psychiatric control subjects. So this suggests that the spindle deficit is not due to drugs and may have some specificity to schizophrenia. Work from our lab bears this out. We had the good luck to study an archival sample from Pittsburgh of early course antipsychotic naive patients, and we find a significant density decrease in the early course patients. Interestingly, when we break them into those who had schizophrenia versus other psychoses, it's only the patients with schizophrenia who show the spindle deficit. And we showed something very similar with spindle amplitude. The spindle deficit in this group, whether you had schizophrenia or another psychosis, correlated with that executive function measures. The more spindles you had, the faster you performed on Trails B, which involves task set shifting, and the fewer errors you made on the Wisconsin card sort test. So it's relevant to cognitive function. We also were able to show a spindle deficit in a small group of non-psychotic first-degree relatives of patients with schizophrenia. And regardless of which group you fell in, whether you were a control, whether you were a patient with schizophrenia, whether you had another psychosis, or whether you were a relative, spindle density and amplitude both correlated with IQ. And this is consistent with the large basic literature showing that spindles correlate with IQ, measures of learning ability, and both procedural and declarative memory consolidation. Another group has replicated our findings. They have found a sleep spindle reduction in both schizophrenia and healthy first-degree relatives of patients with schizophrenia. They also found that it was associated with impaired cognitive function and suggested that it might be an endophenotype, an intermediate phenotype, or an endophenotype. In endophenotypes, in this case, are physiologic markers that are thought to be more closely related to the genes that predispose to illness than are the symptoms that are used for diagnosis. And so far what I've shown you is that there's reduced spindle activity in chronic medicated patients with schizophrenia, but also in early course antipsychotic naive patients with schizophrenia, but not other psychotic disorders, suggesting that the spindle deficit is not due to the drugs that we use to treat schizophrenia, and it's not a general feature of psychosis. Instead, the spindle deficit may be an endophenotype of schizophrenia that predates its onset, persists throughout its course, and impairs cognitive functions. And so the question is, can we target spindle physiology to treat cognitive deficits? And I think the answer is yes, but we need to understand the mechanisms and what we're treating and targeting. So let me just tell you a little bit about spindle mechanisms. There's a large literature from animal studies, and so these mechanisms are relatively well worked out. Spindles are initiated in the thalamic reticular nucleus, which is a thin net-like structure strategically located between the rest of the thalamus and cortex to modulate the interactions of thalamus and cortex. The TRN is the major inhibitory nucleus of the thalamus. It's comprised entirely of GABAergic neurons. And for the purposes of this talk, it has two important functions. During sleep, It powerfully inhibits these thalamocortical neurons, and this results in the burst firing that gives rise to spindles. And during wake, it powerfully inhibits these same thalamocortical neurons to gate the relay of sensory information from thalamus to cortex. So it plays a role in spindle initiation and gating, sensory gating. The TRN is abnormal in schizophrenia. A group from McLean has shown reductions in parvalbumin expressing TRN neurons in both schizophrenia and bipolar illness compared to controls. And there is also a reduction in perineuronal nets, which are extracellular matrices that nurture and support and modulate TRN neurons. So they serve a protective function. So what might we expect if, as a result of an abnormal TRN, there was reduced inhibition of thalamocortical neurons in schizophrenia? One possibility is that there would be increased and less filtered relay of sensory information from thalamus to cortex. And this might result in a fragmentation of attention, which is a hallmark feature of schizophrenia and is captured in this patient's self-report. Recently, I've noticed that noises all seem to be louder to me than they were before. I notice it most with background noises that are always around but you don't notice them. Now they seem to be just as loud and sometimes louder than the main noises that are going on. It's a bit alarming at times because it makes it difficult to keep your mind on something when there's so much going on that you can't help listening to. We might also expect a deficit in sensory gating. Sensory gating can be measured by the P50 ERP to auditory stimulation. And when healthy people have an auditory stimulus and an identical one that follows it close in time, the response to the second stimulus is attenuated. Patients with schizophrenia and their healthy relatives show an attenuation of this deficit, of this, they fail to attenuate the second stimulus. It's like they're failing to gate out this redundant information. So that's a sensory gating deficit. A failure of inhibition might also be reflected in increased communication between the thalamus and the cortex, and it could account for the very well-replicated finding of hyperconnectivity between the thalamus and sensory motor, and auditory cortex in schizophrenia, as measured by resting state functional MRI. So we replicated that finding in this group that also showed a sleep spindle deficit. So, and importantly, these two deficits were correlated. Thalamocortical hyperconnectivity, the greater the hyperconnectivity, the fewer spindles people had. So consistent with the hypothesis that both of these abnormalities are reflecting a common pathophysiology, reduced TRN inhibition of thalamocortical neurons. Both thalamocortical hyperconnectivity and spindles correlate with positive symptoms, which makes sense if you understand hallucinations as a failure of the TRN to gate the relay of internally generated sensory information to cortex. You could have these abnormal experiences. And so what we're seeing is correlation with temporal cortex, and this is a region of temporal cortex that is activated when you hear human voices as opposed to environmental sounds, and it correlates with hallucinations. And in both our hands and in Fabio Ferrarelli's group, we find that the greater your positive symptoms, the fewer spindles you have. So in the literature, thalamic hyperconnectivity is found with motor and sensory motor regions. And this selectivity could reflect the organization of thalamocortical circuits. which can be divided into pathways involving core versus matrix neurons. While the matrix neurons project in a very widespread fashion, the core neurons project in a topographically organized way, and they're the main input to sensory motor cortex. So this suggests that thalamocortical hyperconnectivity, spindle deficits, and sensory gating deficits are more likely to reflect a core pathway abnormality. And we may be able to distinguish spindles generated in core versus matrix pathways using both MEG and EEG. So this is nice work from Dimitris in my lab, showing, and as others have shown, that MEG sees about twice as many spindles as EEG, and it detects spindles with a more focal onset that EEG is missing. So it may be more sensitive to focal core spindles. And in the sleep that follows learning, a number of groups have found that there is increased spindle activity in the same regions that were involved in learning, suggesting that those spindles are playing a role in facilitating the sleep-dependent memory consolidation. So we wanted to look at that with motor regions using the MST. So using anatomically constrained MEG-EEG source localization, Jaouet and Dimitris showed in collaboration with Mati and Shiraz that there is muse suppression in sensory motor regions while people perform the MST. And interestingly, in the sleep that followed the MST compared to a baseline nap, there was increased spindle activity in some of those same regions, suggesting that learning was giving rise to increased spindling. In the test that followed the nap, we tested to see if there was improvement. The amount of improvement correlated with spindle activity in the SMA, a region involved in the supplementary motor area, a region involved in motor planning. So we're planning to take this paradigm to schizophrenia to determine if they show the same focal spindle increase. At the same time, we'd like to try to treat the presumed TRN dysfunction in schizophrenia. And we started by trying a drug called azopiclone, which is a non-benzodiazepine sedative hypnotic that acts on GABA-A subunits that are preferentially expressed in the TRN. And in a small preliminary study, we found that azopiclone increased both spindle number and spindle density. We went on to a larger study because in that study, the increase in spindle did not come with a memory improvement. So we went on to a larger study that included controls and again found significant increases in spindle density, both in controls and in patients with schizophrenia. And in patients with schizophrenia, spindle density correlated with overnight improvement on the MST, so correlated with sleep-dependent memory consolidation. Sadly, azopiclone, despite increasing spindles, did not improve memory, and in fact, it may have made memory slightly worse in the schizophrenia patients. So what gives? Spindles are a mechanism of memory consolidation. Patients with schizophrenia have deficient memory consolidation, and it correlates with a spindle deficit. So why, if we increase spindles, are we not improving memory in schizophrenia? And I think that the answer is that our model was too simple. Spindles don't act in isolation to mediate memory. They are temporally correlated with hippocampal ripples, which nestle in the troughs of spindles and represent replay, and they occur in the upstates of cortical slow oscillations. And it is this dialogue between these three cardinal oscillations of non-REM sleep that is thought to transfer memories from a temporary store in the hippocampus to more permanent representation in the neocortex. And so if this model is correct, what it means is that in order to improve memory in schizophrenia, we have to not only increase spindles, but we have to either preserve or enhance their coordination with these other critical oscillations, ripples and slow oscillations. So in our next study, we looked at the coordination of spindles and slow oscillations in schizophrenia. So this is a canonical slow wave in controls and patients. And what I'm showing you is spectral power in the 12 to 15 range, which corresponds to spindles in controls and patients. And it's a little bit weaker in patients just because they have fewer spindles, but the timing is identical. The spindles activity is clustering in the upstates of slow waves. And in fact, the timing was pretty close. This is, the spindles were occurring in the exact same phase of the slow waves in both patients and controls. So timing looks to be normal in schizophrenia. And in schizophrenia, this timing was important for memory. Both the phase, patients whose spindles peaked latest in the slow wave upstate and were most consistent in this timing showed the greatest overnight improvement of the MST. So the timing was critical. So both spindle density led to memory improvement, the timing of spindles, both the phase in the slow oscillation and the consistency of the timing, and together, density and timing accounted for significantly more variance than either density or timing on its own, suggesting that they're both critically important. So what about hippocampal ripples? So work from Matt Wilson's lab has shown that as a rat is learning a new environment, cells in the CA1 of the hippocampus, place cells in the CA1 of the hippocampus fire as it moves through space. This firing is recapitulated only faster during sleep, during ripples. So ripples are equaling memory replay. So they're playing an important role in replay. Unfortunately, we can't see ripples on the surface on surface EEG recordings or MEG recordings. And so we are collaborating with Matt Wilson and Carmen Varela at MIT because they can measure activity from all three regions at once. They can measure from the cortex, the thalamus, and the hippocampus, and they can look at these three oscillations in the same rodent simultaneously. And this is very useful for evaluating drug effects on the coordination of oscillations and memory. And what they found using this rodent model and azopiclone is that azopiclone actually decreased ripple power compared to vehicle in the rodent model. So maybe that's why azopiclone wasn't working. And wouldn't it have been nice to know this before embarking on a large and expensive clinical trial? So we are now collaborating with them in developing a pipeline to evaluate drugs before we move to clinical trials. And aim one is to evaluate the effects of drugs on the coordination of all three oscillations and memory in the rodent model. And then we can take the most promising agents and bring them to a human study where we can evaluate their effects on memory and the coordination of spindles and slow waves. So how do we prioritize which drugs to test in this pipeline that we're developing? We're looking to genetic studies to begin to find clues to the mechanisms of the spindle deficits. As a prelude to genetic studies, Sean Purcell characterized sleep spindles in 11,630 individuals. It was a heroic study from ages 4 to 97. And he found that spindle density and other spindle phenotypes were very highly heritable, consistent with a large genetic contribution. And just to illustrate this, I'm showing you spectral power plots from MZ twins. These are black females, 11 years old. And what's remarkable is the overlap in the spindle frequency range. I mean, these lines look almost identical, and it's caused these electrophysiological sleep markers to be called a fingerprint. And this is compared to two to demographically matched but randomly selected unrelated individuals, just to show the correspondence compared to the variability. Here's just another example with 51-year-old white males. Now, these individuals also have GWAS, and Joe Panagioteropoulou, I always have to read that. That's still, I'm practicing on that pronunciation, from my lab, has been working with Sean to try to identify genetic contributions to spindles. And this is preliminary data that was presented at the SLEEP meeting in June of 2017. And what it's showing is that there are significant relationships between spindle phenotypes and genetic risk for schizophrenia in a subset of this large sample, such that greater genetic risk for schizophrenia is associated with lower spindle activity. And they've now increased their sample size to 6,000 people, and these results are holding up and have become significant for spindle density. And the goal is to be able to decipher the genetic architecture of spindle deficits in schizophrenia. But some of the risk genes that may contribute to spindle deficits have already been discovered. In 2014, the very large GWAS study identified CACNA1I as a risk gene for schizophrenia. CACNA1I is expressed only in the brain and particularly in the TRN, which generates sleep spindles. And knocking this gene out in a mouse model causes a specific sleep spindle deficit. In addition, missense mutations of this gene have been identified in probans with schizophrenia, but not in their unaffected relatives, implicating this gene in both common and rare genetic variation in schizophrenia and suggesting the possibility of an etiologic role. Jen Pan, a collaborator at the Broad Institute, has created these two mutations in a mouse model. And what she finds is that one of these mutations, the R mutation, is damaging. It leads to reduced trafficking of the channel to the surface of the cell membrane and a reduction in the burst firing that is necessary for spindles. And in an unpublished work, in recent unpublished work, they've also shown that it leads to a spindle deficit. And this team is now working to identify compounds that correct channel functioning and ameliorate the spindle deficit. Risk genes for autism spectrum disorder have also been implicated in spindle deficits. Approximately 1% of individuals with ASD and intellectual disability have patch D1 mutations. Patch D1 is selectively expressed in the TRN early in postnatal development, and recording from the TRN in mice with patch D1 deletions has shown reduced burst firing in these mice. And recording from the surface EEG shows a corresponding spindle deficit. There is also reduced TRN inhibition of the lateral geniculate nucleus of the thalamus. And this corresponds to increased vulnerability to visual distractors. So suggesting that the TRN is failing to inhibit LGN relay of distracting visual information. And importantly, both the spindle deficit and the attention deficit can be rescued by compounds that correct the channel functioning. So how might genetic hits early in development predispose to neurodevelopmental disorders such as autism and schizophrenia? Rodent studies are beginning to provide some clues to that. During gestation, all axons that pass from the thalamus to the cortex have to go through the TRN, which helps to guide them to their terminations. And as early as in the first postnatal week, spindle bursts, which are a precursor to adult sleep spindles, begin to appear and play an important role in connecting the thalamus to the cortex, connecting sensory motor regions of the thalamus to the cortex. So these and other studies are documenting the role of the TRN and spindle activity very early in development in establishing and refining thalamocortical connectivity. So if genetically mediated TRN hits, abnormalities disrupt the development of this circuitry, it could predispose to schizophrenia and other neurodevelopmental disorders. So to summarize, how am I doing? I'm good. To summarize the story that I've been trying to tell you here, the aim of this highly collaborative research program is to try to forge empirical chains, empirical links and causal chains from genes to diagnosis. And the ultimate goal is to try to identify mechanisms that we can treat. So the research program started with a cognitive study showing impaired sleep-dependent memory consolidation in schizophrenia. And this and subsequent work using declarative memory and other procedural tasks has established an impairment of sleep-dependent memory consolidation among the cognitive deficits of schizophrenia. This deficit occurs in the context of reduced sleep spindles, and importantly, the spindle and memory deficits are correlated, suggesting that reduced sleep spindles impairs sleep-dependent memory consolidation. The sleep spindle deficits implicate the TRN and related thalamocortical circuitry in schizophrenia, and reduced TRN-mediated inhibition may contribute to thalamocortical hyperconnectivity and also to sensory gating deficits, which may in turn contribute to positive symptoms. Genetic studies have identified CACNA1I as a risk gene for schizophrenia, and knockouts and mutations of CACNA1I in mouse models impair channel function and give rise to spindle deficits. And so CACNA1I is obviously only one of many genes that give rise to spindles, which are a complex phenotype, and may contribute to the thalamocortical circuit dysfunction that underlies spindle and gating deficits, memory deficits, and symptoms in schizophrenia and possibly other neurodevelopmental disorders, leading to a cascade that could give rise to schizophrenia. And the hope is that this sort of cross-disciplinary research, by helping us to understand these links and these sorts of chains from genes to diagnosis, will give rise to new treatment targets that are mechanistically driven. So with that, in closing, I'd like to thank the army of people who contributed to this work, and this is an incomplete list, and also these are people who have been in mine and Bob's labs over the years, and also our collaborators' labs, and also to our funding sources and participants. And none of this work would have been possible without the CRC, which was a free resource when I just started this program, and as an unfunded labor of love. So I will stop there and happy to answer any questions.
Thank you very much. That was great.
Thank you.
Any questions from the audience?
I stupefied everybody.
Thank you so much for so great presentation. Did you look also circadian disturbances in addition to sleep disturbances in the sample?
We have not, but others have, and there are certainly circadian disturbances. I think what's unclear is whether they're true circadian disturbances or whether it's something about the lifestyle of having a major mental illness that gives rise to the circadian disturbance. They're unemployed, they don't have the same structure during the day. Patients with schizophrenia tend to have a shifted circadian rhythm. Yeah, but we have not formally evaluated that or how that might interact with the spindle deficits. Can't hear you, I'm sorry. Oh, thank you. Sorry, I'm having trouble hearing you. Can you stand up and yell?
Question about why you showed an example of ADHD. Learning deficiency in some cases.
In autism,
Yeah. So
in mice with autism,
right.
slightly different.
Oh, I'm sorry. Yes. In my study, just wondering, is there any difference in ADHD and what that the reason?
So I think there is some genetic overlap between schizophrenia and autism. But what I was trying to show was that it may not, the spindle deficit may not be unique to schizophrenia. There is some signal in the autism literature showing spindle deficits. It's a very inconsistent literature. We've just begun to study that. And that neurodevelopmental disorders may have spindle deficits in common as a potential hit to these thalamocortical circuits that may be important in the expression of both schizophrenia and autism. So that was my point in showing that other risk gene. Tom?
So you might have mentioned it, but if you did, I missed it. Slow wave sleep by itself, has that been studied in schizophrenia? Is there a normal, you talked about the ratio, the timing between spindles and slow wave sleep.
Mm-hmm.
How about slow wave sleep in its absolute magnitude? Is that normal in schizophrenia? Do you know? And has that also gotten studied as an endophenotype?
Yeah. So in terms of the duration and proportion of individual sleep stages, in chronic medicated patients, that looks really normal. So in slow wave sleep, you see these slow oscillations and slow waves in delta power, both in N2 and N3. We have not seen, and others have reported reduced delta power, but it doesn't hold up in meta-analyses, and we have not seen it in our data. So to our eyes, slow wave sleep is looking, the slow oscillations and slow waves are looking normal. Demetrius, would you agree with that?
Yeah.
Yeah. Okay. Yeah. So there may be some specificity to the spindle deficit. In other words, the slow waves are thought to have cortical origins. Archer?
So I'm still trying to understand, like, what the spindle meaning. I understand,
Is this an existential question?
Well, no. Does it correlate with the cellular activity in the GI and the neurons? And the variation over time in the spindles is presumably generated somewhere else. Like where does that happen? Is it sort of a net property of the network or something like?
So, we just had an in-service from Demetrius in our lab about spindle mechanisms, and, despite the bulk of research that's been done on them, it's still not completely worked out. But the TRN, when cut off from both the cortex and the rest of the thalamus, can generate spindle activity in complete isolation. But cortical feedback to that loop, there's collaterals from the corticothalamic neurons that go to the TRN seem to be necessary to synchronize the spindles across the cortex. So I don't know if that begins to answer your question. You're shrugging. Can I?
Well, I mean, these answers are no.
Yeah. It's unclear what is initiating and turning off spindles. Exactly. It's a circuit, and so, people have tried removing different parts of it, but I don't think the answer is abundantly clear.
So very interesting what you found that with the medications that where you saw that when spindles were actually increased, it did not correspond to an improvement in reconsolidation. But I'm curious what else may have changed in the process. Do you see any other variables that were correlated with
So that's what we're looking at now. So I mentioned that hippocampal ripples may have changed, but Will Kuhn in my lab has been looking at spindle coherence from one electrode to another, and finds that there's a slowing that happens under a zopiclone. It also seems, Dimitris has shown that it seems to flatten slow oscillations and make them longer. So it's screwing up sleep oscillations. I think that these meds that we take for sleep have all sorts of untoward effects that we don't understand. And I think using them as pro-cognitive, trying that might have been a bit premature. So, we're also considering transcranial stimulation approaches, auditory stimulation, because I think these endogenous oscillations are quite smart and precisely coordinated, and our drugs are sledgehammers that are not precise. So I don't think there's going to be an easy drug answer here, unfortunately.
Did you find any other consequence of, although did not improve the memory consolidation of schizophrenia with the drug, saying that spindles changed in terms of the number or the deficit there? Were there any other behavioral manifestations?
We only did 2 nights of the drug, so we couldn't really look at symptoms. And we really focused on the MST as our one test and not having others interfere with it. We're interested in looking at the effects of gating as well. And so we've added that to studies that we're now doing to see if the drugs that affect spindles also affect sensory gating and MST. So we are trying to broaden it in the ways that you're suggesting.
One question. My brother-in-law is basically a professional marathon runner, and he's having this transcranial direct stimulation during night to improve his condition. I don't know how he does that, but he says that he's dreaming a lot, much more than before, and I wonder if it has an impact in terms of these symbols and how basically the REM or non-REM phases are doing this. It might be kind of a potential solution, or sort of schizophrenia and other neurocognitive disorders like Parkinson also starts many years before patients having trouble.
And Parkinson's with cognitive deficits is associated with spindle deficits. Yeah. So I think there's a lot of people are coming up with commercial applications of sleep stimulators that are increasing slow waves to try to deepen sleep and improve memory. I think the literature is a bit sketchy. It could be that your cousin is remembering his dreams more because the stimulation is waking him up, and you tend to remember your dreams when you're woken up, otherwise you forget them. I think it's the Wild West out there. Brian Baxter has reviewed that literature with us, and it's really it's very premature, I think, to start stimulating as having commercial applications. I've seen a website where they want to stimulate slow waves in children to old age to improve memory, and I think they're particularly focusing on the dementia market, and it's a little frightening.
Basically, the conclusion is you need to sleep more and better. And I hope to keep it around here because he's always criticizing me for it.
Who's criticizing you? I can talk to them. I'll have a word with them.
Well, let's thank the speaker once again. It's really amazing.
Thank you.
Yeah, so do you think they have, you have to kind of like the same kind of process people tolerant in Parkinson's?
Yeah.
My father-in-law was Parkinson's.